Okay, so just to recap what we learned in the previous video, we talked a lot about nucleotide and nucleotide structures. And I put up this slide here at the end of the last lecture. And I'm showing it to you again because you really need to internalize the information that's on this slide so that you'll be able to understand what we're talking about when we're talking about different nucleotides and different nucleobases and 5' phosphates and all of these things. And so if you just take the time to internalize the contents of this slide, that's going to be super helpful. And again, I encourage you, if you're having a hard time or it feels like too much memorization, check out the companion video and I'll just show you how I have done it over the years, and maybe it will work for you, maybe it won't. Hopefully you'll find it helpful. But so now that we've got the basic structure, chemical structure of the monomeric building blocks of nucleic acids under our belt, we can start to ask questions about the polymers. So how does the cell actually put these nucleotides together to build nucleic acid polymers? And again, what is it about nucleic acid polymers that makes them such ideal carriers of genetic information? What makes them easy to replicate, transmit information, store information, so on and so forth? And we're going to learn the answers to all those questions as we go. So the first thing we need to realize is that nucleotides are joined together to form nucleic acid polymers through phosphodiester bonds. And so if we look at the connection between two nucleotides here in a nucleic acid polymer, what we will see is that there is a connection between the 3' position of one nucleotide and the 5' position of the other one. And the connection that bridges the two together is what is called a phosphodiester linkage. That is, the 3' hydroxyl connects to a phosphate, which connects to the 5' hydroxyl, or what was the 5' hydroxyl of the next nucleotide. And so these 5', 3' phosphodiester linkages are what are the glue that's connecting and holding these nucleic acid polymers together. So how biochemically and chemically are the nucleotides actually joined together in this way? And in the cell, nucleotides are added to the 3' end of a nucleic acid polymer using a high energy nucleoside triphosphate as the substrate for addition. And so if we were to think chemically what's going to happen here, you have some nucleic acid polymer, 5' end working our way down, and this is the final 3' nucleotide that's part of the chain. Well, this hydroxyl can perform a nucleophilic attack on the nucleoside triphosphate you see here. Whoops, pardon me. And in order to make this reaction go, we need to use a base to catalyze and activate this nucleophile to promote this attack. And once the once the 3' hydroxyl attacks this phosphate, it can kick off these two phosphates that are joined together. This is something called pyrophosphate. It's a very high energy leaving group. And the result of this biochemistry is that you will have created a new phosphodiester linkage between the original nucleic acid and your new monomer here. And I just want to emphasize that this the 3' hydroxyl of the nucleotide that we added now becomes the new 3' end of the molecule. And so from this point of view, we can say that nucleotides are joined together in the 5' to 3' direction of the polymer because they're always added at the 3' end. So it's really important for you to know the chemical mechanism here, and basically we can sum it up in sort of 3 steps. You need to know that there is base activation of the 3' hydroxyl of your chain. This allows for nucleophilic attack of the 3' hydroxyl on the alpha phosphate, that is this first one here, of a nucleoside triphosphate, and then the pyrophosphate can then act as a leaving group to drive the reaction forward. And again, the other key takeaway is to know the polarity of this reaction, which is that the nucleic acid polymer is growing in the 5' to 3' direction. That is, new nucleotides are always going to be added at the 3' end of the polymer. And that's really important. Okay, so we've connected a whole bunch of nucleotides together to make a polymer. Woo, cool. So what? So we've got this big, long polymer of nucleotides and nucleobases. what is the purpose of this, or why should we care about these polymers? Why do they have genetic information? Well, things start to get really interesting when we recognize that the individual nucleotides that make up the polymer can interact with one another through what is called complementary base pairing. And the core concept to understand this complementary base pairing is the notion of a hydrogen bond. And so you might remember from earlier in the class or in other chemistry courses that if you have an electronegative atom that has a lone pair, and you have another electronegative atom that has a hydrogen attached to it, an acidic proton, then the proton has some partial positive character, and the lone pair is going to have some partial negative character, and an electrostatic interaction can occur between these two groups, and this is what is called a hydrogen bond. And these types of hydrogen bonds are basically the fundamental interaction that's going to drive a lot of the complementary base pairing that we observe. And so you should know that there are hydrogen bond donors. So these are things that have the proton attached to them. And so those include things like a primary amine or hydroxyl group. And there are hydrogen bond acceptors. These are the things that have some sort of lone pair, which can basically serve as the acceptor. So these are things that are going to interact with donors to form hydrogen bonds. And so if we take a closer look at in this case, the 4 DNA nucleobases, and we look at the structure of those rings, you'll see that, okay, if we look at adenosine, we've got primary amine groups. So there's one donor, which we can denote by this arrow pointing there. And we have nitrogen on the ring here that's got a lone pair, so that's an acceptor. And if we look at cytidine, we see that we have primary amine, so that's a donor. We have the lone pair on this nitrogen, so that's an acceptor. And we've got a carbonyl group, so the lone pair there can be an acceptor. And so cytidine has 3 potential hydrogen bond interactions that can form. If we look at the guanosine, we can see that it has a carbonyl here, so that's a lone pair is going to function as an acceptor. We've got a nitrogen here with a proton on it, so this can function as a donor. And we've got another amine here, so that's a second donor. So there's 3 possible interactions, hydrogen bonding interactions that can form here. And if we look at the thymidine, then we see again a carbonyl, so the lone pair there can be a acceptor. We see this NH, and so this here provides a donor opportunity. And we see another carbonyl here, and so this is an acceptor. So there's 3 possible interactions that thymidine can form. Okay, so each nucleobase then kind of has its own little repertoire of donors and acceptors. And as I said before, this can allow the different nucleobases to interact through one another through these hydrogen bond donor acceptor things. And one of the famous things that Watson, Crick, and Franklin realized when they started to look at these interactions was that there are very specific base pairing interactions that can occur between certain combinations of the base. So if you've never seen this movie, Life Story, Race for the Double Helix, it's an interesting, albeit highly biased, dramatization of the discovery of the DNA structure. Jeff Goldblum plays a very funny James Watson, but there's a sequence where he's playing around with each of the nucleobases and what hydrogen bonds they can form, and eventually he recognizes that there's these beautiful base pairing interactions that they can form. And we can kind of do this for ourselves if you were to say screen cap the different bases I just showed you and then start rotating them around and flip them. I'm sorry that some of the letters here are flipped, but it's a nice way to show. So if we take the adenine here, which had its 2 donor acceptor pairs, and we take thymine over here, then we can see that the carbonyl here can form a hydrogen bond with the amine over here, and the NH here can form one with the lone pair of the nitrogen here. And so you can get a nice 2 hydrogen bond interaction between A and T. And if you look at, say, guanine instead, we've got our 3 possible interactions here. And as before, we can see that the primary amine and the carbonyl can interact here. We can see the NH and the lone pair nitrogen here could interact. and the primary amine and the carbonyl here could interact. So G and C can form a nice 3 hydrogen bond interaction. And if you try to do this with other combinations, so you say, well, maybe we can make an AC base pair or something like that. If you just draw it out and you try to put them together, you see, well, these got 2 amines pointing at one another. That's not really going to work because these are both donors, right? And same thing here, we've got 2 acceptors. They're not really compatible with one another. And similar, if we try G with T, for example, we can see we've got 2 carbonyls. Those are 2 acceptors. It's not going to work. 2 donors. It's not going to work. Here, maybe we can make 1 base pairing interaction possibly between a G and T, but these other interactions are unfavorable. And so really the primary combinations that work are A and T and C and G, and they differ between making 2 hydrogen bonds or 3 hydrogen bonds. And so what this means is that there's a highly specific complementary base pairing interaction that can occur between 2 strands of DNA. And so the hydrogen bonds involving the amino and carbonyl groups are the primary mode of interaction that can bring 2 strands of DNA together. And in the most famous Watson-Crick-Franklin hydrogen bonding pattern that I showed you on the previous slide, this occurs when the strands are oriented anti-parallel to one another. So if one strand is running 5' to 3', you can form those beautiful base pairs that I showed you if the other strand is running in the opposite direction to it. And under these conditions, as I mentioned before, adenine will form 2 hydrogen bonds with thymine, and guanine will form 3 hydrogen bonds with cytosine. And we typically call these interactions base pairing interactions. And so again, one thing you're really going to want to know is what these Watson-Crick-Franklin base pairs look like. And in fact, it's also going to help you when you're trying to learn the nucleotide structures. If you know which nucleotides pair with which, it can actually help you draw the structures, because you know that you're going to have complementary base pairing interactions to pull these particular nucleotide combinations together. Okay, so what happens if you allow 2 single strands of DNA to interact with one another through these base pairing interactions with one strand running anti-parallel to another? Well, if you do that, then what you get is probably one of the most famous and beautiful macromolecular structures in the history of science, and that is the B-form double-stranded DNA double helix, which was discovered by Watson and Crick and Franklin many years ago, and sort of completely catalyzed our molecular understanding of biological processes. And so if you look at the structure you get, what you find is that you have a beautiful right-handed helix in which one of the strands is running 5' to 3' like this, and the other is running anti-parallel to 3' to 5' like that. And there are these base pairing interactions between the 2 strands, and it takes about 10.4 base pairs to do 1 turn of this helix. Now, if we were to look kind of like down, rotate the structure so we're looking down the center of the helix, what we would find was that the sugar phosphate backbone is completely on the outside of the structure, and all of those nucleobases in the Watson-Crick-Franklin base pairs they form line the inside of this structure. Okay, so it has this very kind of beautiful phosphates out, bases in geometry like you see here. And if we were to look at any one of those base pairs, we would see that indeed they form these kind of canonical base pairing interactions that I showed you on the previous slide. I won't tell you which of these is which, so it's kind of an exercise for yourself. You should be able to tell which of these is an AT base pair and which one of these is a CG base pair. Okay. Now, the most incredible aspect about the structure of DNA to recognize and appreciate is that although the AT and CG base pairing interactions are completely orthogonal, that means A can't pair with C, G can't pair with T. It doesn't work. They're not compatible with one another. Despite the fact that the interactions are orthogonal, an AT and a CG base pair form units that can be stacked perfectly one on top of another to stabilize the helical structure. And again, this was something that was recognized during the structure of this, that you have these units here that are distinct from one another, but if you superimpose them on top of one another, it's an almost perfect stacking interaction. And so what are these base stacking interactions I'm talking about here? Well, we just have a nice representation of each base pairing interaction in a small piece of DNA strand here. And so there are going to be van der Waals interactions between the various hydrophobic faces of the nucleobases. So remember that there's lots of carbons there that occupy space, and so you get these nice van der Waal type effects. And then there's also electronic effects that come from a phenomenon called pi stacking. So remember that all of these nucleobases are highly conjugated structures. So they have electronically, they have all these p orbitals oriented upwards, and those types of structures are very well suited to stacking on top of one another. And so these base stacking interactions have the effect of reinforcing individual AT or CG hydrogen bonding interactions that's going to drive a massive stabilization of the whole double helix itself. And that's sort of the beauty of this structure, that AT and CG are completely orthogonal, but each base pair could fit perfectly on top one of the other. And so the fact that the energetics of this structure demands complementarity between the nucleobases in each strand, that's what's going to provide the basis for genetic information storage, replication, and transmission within nucleic acid polymers. Because if we were to pull these two strands apart, we now know exactly what the other strand should be based on the complementarity between these base pairing interactions. So we know if we separate these strands that it should go G, C, G, C, T, G, T, G, T, C, G, A, just based on this strand alone. Okay? And that complementarity, that is that one strand tells you what the other strand needs to be perfectly, is really why nucleic acid polymers reign supreme in terms of this information. You're hard pressed to find something like that in the realm of polypeptides or lipids or other types of things. It's a really remarkable property, and it's one of the key ones that you should really understand. And of course, in future lecture, we'll actually learn how the cell does this type of replication task and does this copying and things like that. It's very interesting. But conceptually, that complementarity is really all you need to know. Okay, but one other thing that's interesting about B-form DNA to appreciate is that you don't necessarily have to pull the strands apart to know some of the sequence information that's inside. And that's because the B-form double-stranded DNA helix, when joined together, presents two large grooves which provide access to the nucleobases. And so if you look at this kind of space-filling representation here, we've got the sugar phosphate backbone here in green, and you'll see that there's these two grooves that provide some access to the nucleobases inside. You have this very wide, deep groove, which is called the major groove of DNA, and you have this more shallow and narrow groove called the minor groove. And what's neat is if we look at base pairing interactions here, so A and T, we've got these two red hydrogen bonds that form the Watson-Crick-Franklin base pairs, and in C and G, we have the three right there, so these are highlighted in red. What you'll see is that there are additional hydrogen bond donors and acceptors, as well as van der Waals interactions that can be presented on each of the grooves. So this side over here in green are the interactions that are possible with the major groove, and over here in purple, these are the interactions that are possible with the minor groove. And what this means is that this combination of different acceptor and donor patterns presented by adenine, thymine, guanine, and cytosine in their base pairing can actually be partially accessed through interactions with these major grooves. And so what you find is that there are many proteins that can actually bind to double-stranded DNA through the major groove and read the sequence that's inside without even needing to pull it out. So in addition to the two strands coming together being perfectly complementary, the B-form DNA structure also permits access to the information inside without even needing to pull it apart completely. And that's going to end up being really useful for a lot of gene regulation and other phenomena that we'll talk about later in the course. Now, what I will add is that the B-form DNA structure I just showed you, it's sort of the most beautiful and elegant nucleic acid structure in biology, but it's really just the tip of the iceberg in terms of nucleic acid structure. And so in the next video, we'll talk a little bit about alternative DNA structures that can form, a little bit about DNA-RNA hybrid structures that will crop up in various processes like transcription or DNA replication. And there can even be much more elaborate, almost protein-like nucleic acid structures, functional RNAs that have very complex three-dimensional structures. And we'll talk a little bit about how those structures form and the roles they play inside the cell.